Good afternoon, and welcome to the PET lecture in the summer series of imaging of brain imaging. My name is Bob Innis, and I'm the chief of the molecular imaging branch in the intramural program of NIMH. This slide that I have up here is a diagram of what I think my career has been. And then I want to tell you where I want my career to go in the next and presumably terminal phase. But what I will be telling you about is, among two major topics, is imaging of neuroinflammation with this radioligand PBR28. We developed this radioligand. It binds to TSPO, translocator protein. I'll explain that more later, and which is highly expressed in inflammatory cells in the brain and in the periphery. So when we first developed this radioligand, we wanted to see if it would work. And we tested it in a rat that had a stroke. We induced a stroke in the rat. And sure enough, you could see uptake, and it was binding to what was known to be inflammation around a stroke in a rat. Then going up the phylogenetic tree, we go into studying monkeys, and we can uptake and do complete quantitation. So we end up with a number, I won't go into it, but this VT number is proportional to the density of the target sites. We extended it to healthy human subjects, and it shows the distribution made throughout the brain, higher in gray matter than in white matter. And along the way, we got a healthy subject from Hopkins who wasn't so healthy. And the clinical fellow at the time, Chuck Kreisler, said, you know, I wonder if this guy actually had a stroke. And he did. So I think it's clear that this work has been translational, but what I want to try to do in the future is to try to connect it to treatment trials to get two basic pieces of information. One is pathophysiology, and the other, most important, would be treatments. So for example, and with regard to my own specialty of psychiatry, do a subset of patients with depression have neuroinflammation, as shown by PET imaging? And could a drug that inhibits the activation of microglia decrease the PET signal and treat depression? So this would be a novel mechanism, and, well, it's not a novel mechanism, but it would be a novel treatment for at least a subset of patients. I can tell you that the answer to this question, do a subset have neuroinflammation, the answer is yes. I'll show you one of 4 studies that have now shown increased inflammation in a group of patients, in some patients with depression. And as far as the drug that inhibits the activation of microglia, I should mention that this study in depression was funded by Janssen, who has maybe a drug in preclinical development that would inhibit the activation of microglia. So it might be possible to do these sort of studies. Okay, so then the title of my talk will be Positron Emission Tomography of Human Brain Can Monitor Neuroinflammation and Cyclic AMP Signaling. That's the other part of the talk, applications to Alzheimer's disease and depression. But what I mainly want to try to convince you is that these tools, I think, are poised to help in clinical trials in Alzheimer's disease and depression. I think it's sort of easy to see it for the inflammation, have an anti-inflammatory. For the cyclic AMP, it's going to be a harder sell, but I'll try. Okay, so linking this is really what I want to talk about, linking PET to therapeutic trials in dementia and depression. To be sure that we're all on the same page, I'm going to compare PET to the better-known technology of MRI. So the spatial resolution of MRI is much, much, much better than that available for PET. But the way PET shines is that it has great sensitivity. It can detect that radioactive atom down to -10 to the -12th, or 10 to the -14th molar. In contrast, MRI or any MRI technique like MRS, that primary signal must be present at least 10 to the -4th molar. Since many of the receptors that we're interested in are present at 10 to the -9th molar, there's no way in my lifetime that MRI is going to be able to do that. So what we do is connect a radionuclide like carbon-11, which can be detected with high sensitivity, with a ligand, let's say raclopride, a drug that's selective for the D2 type of dopamine receptor. And we end up with a radioligand, which has high sensitivity because of the carbon-11 and high selectivity because of the raclopride. And that's what we'll be doing. So just to summarize, because there's a lot of information going on, what I will tell you is that In Alzheimer's disease, we found that TSPO binding is increased in Alzheimer's disease, but not in mild cognitive impairment, which is a precursor syndrome. The increased TSPO binding correlates with disease severity based upon a cross-sectional study, and also with disease progression based upon a longitudinal study. In major depressive episodes, subjects are currently depressed. The TSPO binding was increased in the unmedicated patients, but the TSPO binding was not changed in those who were on medication. Okay. This target that we're imaging is translocator protein, which was previously known as the peripheral benzodiazepine receptor. It is a protein located in the outer mitochondrial membrane, and it transports cholesterol to an enzyme that synthesizes pregnenolone, which is a precursor for steroids and neurosteroids. It is highly expressed in macrophages, activated microglia, and reactive astrocytes, with the last two being the major inflammatory cells in the brain. We're using it not to study its function, but as a putative biomarker of the activation of the immune system in the brain. So it's kind of something that I will call neuroinflammation. But that's a very loose term, and it's a very broad term. If I say neuroinflammation, what I really mean is increased TSPO binding. So as mentioned, we induced a stroke in a rat, and this was the actual PET image from that rat. And there's an ischemic core in the middle, increased binding of this radioligand that we developed surrounding it. We killed the animal, and we could stain, and we could see the necrotic core. And then to confirm that we're really imaging the TSPO, we took a tissue section and did binding, in vitro binding with controlled conditions, with another radioligand, actually the prototypical radioligand, PK. And we could do this fully quantitatively in the rat, and we confirmed that we could measure the density of TSPO surrounding this necrotic core. We went ahead to monkey subjects, and this shows the uptake. Because of the very high sensitivity of PET, we can inject minute amounts that have no pharmacological doses that might occupy 1% of all of the TSPO in the brain. We can block that uptake pharmacologically with pharmacological doses of another compound, let's say PK itself. This color scale is accurate. More than 95% of the uptake in the monkey brain is specific and can be blocked by other agents acting at this site. So this is a really super radioligand for monkeys. In humans, there's much less binding. That's because there's a much lower density of TSPO in human brain than monkey. But we could do this full quantitation such that the pixel of color here represents the density of the target sites. And this is the expected distribution in the human brain. It's widespread, higher in gray matter than in white matter. But as I mentioned, there was this healthy subject, a man in his 50s with a history of hypertension, who came in and had this bright red spot. And Chuck Cruikshank said, you know, it's almost like a stroke in the rat, but like a bigger brain. And but his screening MRI was completely normal. So we had him go back, see the neurologist at Hopkins. They did another interview, and he said, well, you know, doc, actually, 2 weeks before the PET scan, I woke up in the middle of the night and had some tingling and weakness in my fingers and a little bit of odd feeling in my face. But when I woke up, I went back to sleep, woke up, and there were no symptoms. So then we have this screening MRI was 6 weeks before. He had this mini stroke, lacunar stroke, 2 weeks before the PET scan, and then an MRI afterwards shows on two different sequences this lacunar or small stroke in the basal ganglia. It's just an incidental finding, but it did show, at least in this case, that the PET imaging had sensitivity to pick up a stroke that was missed by a neurologist on history and physical exam. Okay. We wanted to look at this radioligand prospectively in other disorders that have localized increase of TSPO. We looked in patients with epilepsy, for example, where the epilepsy begins in the medial temporal lobe. And in 15 out of 16 patients, there was increased TSPO binding in the epileptogenic focus in the area of the brain where the seizure began. The next major way we wanted to go ahead was to look at Alzheimer's disease, because that's going to be a more widespread inflammation, more typical of what one would expect in, let's say, psychiatric disorders. So neuroinflammation is a proposed contributor to Alzheimer's disease pathology. It is certainly present at the time of death, and it may be involved in the pathology. But it's unclear whether it's an early or a late phenomenon in the course of the disease. Prior TSPO PET studies have shown conflicting results in Alzheimer's disease and in the precursor syndrome, mild cognitive impairment. So we had developed PBR28, which has a much greater specific signal than the prototypical agent C11 labeled PK. I think the major reason for the conflicting results with the prior studies was that radioligand, the one that was first developed, C11PK, has extraordinarily low signal-to-noise ratio. So we use this new and improved one to try to detect expected differences in TSPO density between Alzheimer's disease and controls, and then to find out what it was like in the precursor syndrome, MCI. The initial sample, published 5 years ago now, had 19 AD, 10 MCI, and 13 healthy controls. This and most of my other studies, the major weakness is a limited sample size. It's I think, a deficiency, believe it or not, in the clinical center for many investigators, including myself, to be able to recruit patients. But in any case, that's the sample size. Have the age. This is an overall measure of cognitive impairment. On this mini mental status exam, a 30 is perfect, 28 is not so bad, MCI, and 20 would be typical of moderate Alzheimer's disease. A critical part of this experimental design is that all subjects had to have a screening PET scan for amyloid. There's a PET radioligand for amyloid, one of the two proteins that build up in Alzheimer's disease. And we required that the two patient groups, AD and MCI, be positive for amyloid, and that the healthy subjects be negative. And the reason why we designed that is that you can think of MCI as sort of granddad with a little bit of forgetfulness. But we didn't want that. We wanted something more. We wanted granddad with a little bit of forgetfulness plus a positive amyloid scan. Because a positive amyloid scan is a boatload of amyloid. And those individuals with mild cognitive impairment and a positive amyloid scan have a high risk of converting to Alzheimer's in the next 1 to 2 years. So the question being addressed here, are these individuals who are at the precipice of converting to Alzheimer's disease different from AD or healthy subjects? The result is shown in these average images, and there was an increase in most areas of the brain in Alzheimer's disease compared to either control or mild cognitive impairment. This color scale is accurate. If you wanted to look at a representative region in the neocortex, the inferior parietal cortex, you can see that the increase in AD compared to healthy controls or MCI at the 0.001 level, but no difference between MCI and healthy controls. We also looked at the cerebellum, which is minimally involved in Alzheimer's disease, and there was no difference between those groups. So if you wanted to look at the regions that were most severely affected, this is shown here, Alzheimer's disease greater than healthy controls, and it's primarily in the temporal lobe, especially the medial temporal lobe, which is the first and most severely affected in early stages of Alzheimer's disease. It's where the tau is deposited, by the way. Perhaps the most important finding in that study, that you'd not be able to do with a postmortem easily, is that there was a correlation between the PBR28 binding and the clinical dementia rating scale, done after you statistically normalized. And the correlation was 0.001. So cross-sectionally, where each subject has one scan, more inflammation, the greater the cognitive impairment. It is a putative biomarker of disease severity. I mention that because there's no correlation between disease severity and the amount of amyloid that's deposited, and no correlation at all. This one does correlate. And that can be understood from this generally accepted schema of the progression of problems in Alzheimer's disease. One of the first things to develop in Alzheimer's disease is actually the deposition of amyloid in the brain. And it reaches a peak and stays about that same level from the beginning of MCI to the end. So the reason why there's no correlation with amyloid is the patient is getting worse and worse and worse, but the amyloid's already been stable. There are various other ones here. This is clinical function, a little bit of deterioration at the end of MCI, and then it goes to hell. This is clinical function. So our marker here, with limited sample size, suggests that this is a marker of clinical function, and it correlates with disease impairment. Okay, then these subjects, the ones we could, were followed up longitudinally, and the idea was to determine if TSPO binding increases during progression of AD and in normal aging. There were patients, AD and MCI, and we did the correction for partial volume errors. It was very clear in the patients there was a loss of cortical gray matter, and we had to account for that. So, what? This is this is backwards. This is backwards. So let's just skip that one. If you look at the results in all of the patients showed an increase in TSPO binding over a 2 to 4 year period of time, whereas the controls showed no increase. There's no increase during normal aging. But again, perhaps the most important finding was that there was a correlation between the increase in the TSPO binding and the increase in the cognitive impairment at the 0.004 level. So it is therefore a putative biomarker of disease progression. This is starting to get quite interesting. So the conclusions from the Alzheimer's study cross-sectionally, 1 scan per subject. Neuroinflammation occurs after conversion of MCI to AD and worsens with disease progression. It is therefore potential biomarker disease severity. The longitudinal study, it increases in AD but not in controls, and that increase in the patients correlates with disease progression. So it is therefore a potential biomarker of disease progression. Okay, we looked at some other forms of dementia. Alzheimer's is fairly widespread in the neocortex, but we looked at 2 others. And one of them here is posterior cortical atrophy. It's a very rare variant of Alzheimer's disease where the degeneration is in the occipital cortex. And as you may know, the occipital cortex is involved in the where and the what information processing. So these individuals will have problems identifying objects and knowing where they exist 3-dimensionally. I might also mention that they have often can have visual hallucinations. So we compared patients with PCA to amnestic AD. Amnestic is the memory impairment AD, the typical type, and controls. And they were imaged with a radioligand for TSPO, amyloid, and then just FDG. What I want to show you here is the increase in the TSPO binding. I've shown this to you previously. This is Alzheimer's disease versus healthy controls. The greatest increase is in the temporal lobe there. But in posterior cortical atrophy, it's in the posterior cortex where you might expect it to be. These studies are not ours, but these studies are done externally and looks at the deposition of a radioligand for tau protein. And you can see that where the tau is in PCA and AD correlates where the inflammatory changes are. So this mirrors more the distribution of tau in the brain, much more than amyloid itself. Okay, having looked at a disorder where there's a known neuropathology, where there's, we decide, and the radioligand does work there, consistent with the known pathology at death, we went into a psychiatric disorder where there's no known neuropathology, and it's unclear whether neuroinflammation is involved. However, I can say that there's a lot of evidence suggesting that at least a subset of patients have their depression caused by inflammation in either the brain or in the periphery. As a disclosure, as mentioned, this was supported not only by NIMH, but also by Janssen, which had, or maybe still has, a drug in early development to inhibit the activation of the inflammatory cells. The study aims were to evaluate TSPO binding in major depressive episode. Major depressive episodes means they're currently depressed compared to healthy volunteers. And we wanted to investigate the effects, if any, of medication on TSPO binding, and therefore half of the patients were on antidepressants and half were not. The distribution of the subjects is shown here, healthy volunteers 20, and then, mind you, all of the patients are all depressed. Some of them were on medication, some of them were not. These are symptom rating scales for depression and anxiety, and these symptom rating scales would be consistent with a moderate level of depression. What we found is that there was a widespread increase of TSPO binding in the MDE patients, not the non-medicated MDE, that should be non-medicated MDE compared to controls. And this was in all regions of the brain, including the cerebellum. A representative distribution is shown here in the anterior cingulate with this fully quantitative measure. And you can see that it's increased in the unmedicated, but there's no difference between the medicated and the healthy subjects. As far as a difference, it's about 30% higher on average. But of importance here, there are some subjects that are highly rated up here. We have to remember that depression is a heterogeneous disorder. There's going to be some people with depression, I'll bet, caused by inflammation, some by cyclic AMP signaling, some by genes, and so they're all different types, but we don't know how to break them out yet. But this may be, if I were going to give an anti-inflammatory, I would go after that group right there and compare them with the lower group down here. Okay. The major finding is that TSPO binding showed a widespread increase in unmedicated MDE patients compared to controls. This actually replicates the first finding by Meyer et al. In 2015, and we began about the same, sort of about the same time, but we were slow in getting the patients, which is a problem for us, and also slow in writing up the paper. So this paper was accepted last month, and with its publication, it will be the 4th study showing increased TSPO in unmedicated MDE. I can tell you it's very rare to have a replication in psychiatry and almost unheard of to have replication of PET in psychiatry. So this may actually be true. But the medicated MDE patients, and this had not previously been looked at, showed normal TSPO density. There are various potential explanations for why that would be the case, but it could be that when they were treated, they were on an SSRI. The SSRI may modulate this PET inflammatory marker, may decrease it. So if there are further studies are being planned and are being done, you're going to have to control for the effect of the SSRI, which may itself decrease this biomarker of prior inflammation. What we really need to address that issue would be a longitudinal study in patients before and after treatment, and I understand that Jeff Meyer is planning to do that. Okay, in summary then, for this portion of the talk, translocator protein is a marker of inflammation. It's highly expressed in activated microglia, reactive astrocytes, and macrophages. In Alzheimer's disease, we found increased TSPO binding correlates with disease severity based on the cross-sectional study and with disease progression based upon the longitudinal study. And in major depressive episode, 4 studies have now found increased TSPO in major depressive episode while patients are currently depressed. So how can PET facilitate anti-inflammatory trials in depression and dementia? Because this is where I want to go. We could not use the TSPO drugs. All of the TSPO ligands increase the transport of cholesterol and increase the production of cortisol. So if you gave a TSPO drug, it'd be sort of like giving prednisone. And prednisone is far too gross or coarse a drug to be useful in disorders like Alzheimer's disease or depression. So steroids may not work, but non-steroidal anti-inflammatory drugs are worth trying or trying again, in my opinion. The non-steroidal anti-inflammatory drugs like aspirin inhibit COX-1 and/or COX-2. The COX system is shown here. COX-1 and COX-2 make the same prostanoid, PGH2, from arachidonic acid. And then its effects depend upon which cell that COX-1 or COX-2 is located in. So the subsequent metabolic machinery there is how the PGH2 is transformed into these final prostanoids. And they can have exactly opposite effects, and I always get it wrong. But if you inhibit the COX in the platelet, you will decrease thrombosis. If you inhibit the COX in the endothelium, you'll increase thrombosis. So you can have exactly opposite effects, and it depends very much on what cell it's located in. And I should also mention that COX-1 is constitutive. It is present in cells at baseline, whereas COX-2 is hardly present at all at baseline, but it is markedly upregulated or induced by inflammation. We developed 2 radioligands that are highly selective for COX-1 and COX-2. They're potent and selective, as shown here. High potency for COX-1, very impotent for COX-2, and contrawise for this other radioligand. So, and I want to mention, again, COX-1 is constitutive, it's hardly affected by inflammation. COX-2 is highly inducible. So with regard to its cellular localization, which is going to be very important for function, COX-1 is primarily located in microglia, whereas COX-2 is present heavily in neurons and less so in microglia. Based upon this just cellular distribution, if I were going to inhibit one of these for anti-inflammation in the brain, I would go for COX-1. And there's further evidence to support it too. In any case, here's the COX-1 radioligand studied in the monkey brain. So there's uptake, and it can be blocked by other compounds selective for COX-1. So it works well. There's a high distribution, including in the pituitary for COX-1. This is the first successful COX-1 radioligand to image COX-1. COX is listed, is present in the periphery, not just in the brain as mentioned here, but also in the spleen, the kidney, and the GI tract. And it's in fact the inhibition of the COX-1 in the GI tract caused by aspirin that is responsible for the GI irritation of those NSAIDs, non-steroidal anti-inflammatory drugs. And you can see the specificity by looking at the blockade with pharmacological doses of the non-radioactive compound. So we have extended this to healthy subjects within the past year. This was the first subject, and these subjects are having 3 scans. They have a baseline scan, then they have one with aspirin, which is a little bit COX-1 preferential, and following celecoxib, which is highly COX-2 selective. And we're getting results that are consistent with the expectations of the pharmacology in humans. So for COX-1, it was easy. We could image COX-1 because it's present there, and we can test its specificity by giving drugs acting at COX-1 or COX-2. For COX-2, we had a really big problem because at least in the monkey, healthy monkey, we could not detect any specific binding, displaceable binding, except in the ovary, which is shown, marked with the arrows here, at baseline and after blockade, and also on the CT. And it's known that the ovary does have a very high concentration of COX-2, but no place else. So in order to test whether the radioligand was really working, we needed to upregulate it with some inflammatory challenge. And so we injected intracerebrally into the brain, into the putamen, LPS, an inflammagen. And we see an upregulation of about 50% of that binding, but we can go to that quantitation a little bit later. And that uptake can be blocked with COX-2 compounds, but not by COX-1. So it was working in this model. I won't show you the data, but there was no effect of the LPS on COX-1. It was not inducible in the brain, whereas COX-2 is. The inflammation, so we looked at the mRNA levels, and if you look over here, this is color-coded. I'm partially colorblind, so I don't quite see it. But those who have better sight tell me that it clearly shows that the COX-2 mRNA is located in the neurons. This one I can understand. So before LPS, there's virtually no COX-2 mRNA in the brain, but a big increase in COX-2 after the LPS, because we sacrificed the monkeys and euthanized the monkeys and looked at that. And I can say also that the majority of it, 80% of it, was in neurons. Okay, we wanted to have an even bigger effect, so we decided to inject the monkey a second time. So we injected the monkey a second time, and then there was a really dramatic increase, anybody can see that, in the putamen. And we could block the uptake as shown here. Those who have good eyes can see that, well, this was a little odd, because when we did the blockade study, there was less uptake here. After you block all the specific binding, then what's left is the nonspecific binding. So why did LPS decrease the nonspecific binding? Well, we sacrificed the animal, and basically what we found is that the second injection had caused like a necrotic lesion. Most of the brain was gone, not all of it. and it was heavily infiltrated with neutrophils. And neutrophils express high amounts of COX-2. So what we're seeing here is the COX-2 expressed in activated neutrophils that are coming into this region. Okay. The conclusions are that PS13 selectively binds to COX-1, which is constitutively expressed in brain, spleen, GI tract, and kidney. Neuroinflammation does not increase its expression. Whole body and brain imaging studies are ongoing by Minjong Kim. MC1 selectively labels COX-2, which is inducible by neuroinflammation. And the majority of that increase is in neurons, at least following the first injection. We want to try to test this radioligands in human subjects. We're not expecting much in healthy, so therefore we hope to start a study this fall, it's almost approved, in rheumatoid arthritis and myositis, which have marked upregulation of COX-2. And we'll find out whether the radioligand works in human subjects. If it does, then we'll go and consider doing this, let's say, in Alzheimer's disease, which has an upregulation of COX-2 postmortem. Okay, and the COX-2 mRNA and the protein, I didn't show you that, are upregulated in inflamed brain and located primarily in neurons. So, and let me just mention something about the time course. So TSPO is slowly responding. COX-2 is rapidly responding. So the TSPO can increase after, let's say, damage in the brain, over maybe 3 days before it reaches a peak, and then can stay there for a long time, and it can stay there for life. In contrast, COX-2, both at the mRNA and the protein level, it's an immediate early gene, can go up and come down within a couple of hours. So as far as using these two radioligands or two different targets, what I put together in my own mind is if you want something that represents a long history, then use TSPO. If you want to look at something acute, you're going to have to do COX-2. The way I explain it is that hemoglobin A1C, right, it's the glycosylated form of hemoglobin, and it reflects the amount of glucose that's been present in the weeks or months prior to the sampling. But if you want to know the immediate effect of insulin on glucose, you're going to have to measure plasma glucose because there'll be no immediate change in the hemoglobin A1C. So if you wanted to have a measure of chronic inflammation over the past weeks or something, use TSPO. If you wanted to look at an immediate changing effect, you would look at COX-2. I might mention that there are some studies ongoing now here in Miles Herkenham's lab, which is showing that stress in rats increases COX-2 in the brain very rapidly. Okay, I'm going to come back to how I try to connect that to therapeutic trials, but let me try the more difficult one now here, and that's looking at the cyclic AMP cascade in major depressive disorder. What I will tell you is that the cyclic AMP cascade is downregulated in unmedicated patients, and it is upregulated with treatment. So an outline of what I'll tell you. The in vivo binding of the C11 labeled rolipram. Rolipram is a reversible inhibitor of phosphodiesterase 4. PDE4 is the major enzyme in the brain for metabolizing cyclic AMP. The binding reflects the activity of the cyclic AMP cascade because of a feedback mechanism that's known from in vitro studies. Increased cyclic AMP stimulates protein kinase A, pKa, which phosphorylates PDE4 and increases its enzymatic activity, and coincidentally, increases its affinity for rolipram binding about 10-fold. So if you have too much cyclic AMP, the body responds with a negative feedback via pKa, increasing the activity of the enzyme that will metabolize cyclic AMP. I'll show you some data on that. What we found, and this had a good sample size, I trust it, rolipram binding was 18% lower in unmedicated patients with MDD than in controls, indicating a downregulation of the cyclic AMP cascade in major depression. The SSRI treatment, consistent with numerous animal studies, increased the rolipram binding by 13%, suggesting a normalization of the cyclic AMP cascade. So this is a good diagram to start with, coming from Ron Duman and Nestler, when the three of us and others were there at Yale. shows, as an example, serotonin and norepinephrine acting at the cognate receptor via a coupled to a G protein to increase cyclic AMP, which activates PKA, which can phosphorylate Krebs, and it goes into the nucleus and leads to neuronal plasticity and cell survival. So what we're going to be doing is imaging this enzyme here, PDE4, with Rolipram, because it's a reversible inhibitor of Rolipram, of PDE4. So we're going to be imaging this portion of the cyclic AMP signaling cascade. And this is the thing that's a little difficult to understand because it's opposite to what you might think. But enzyme activity and Rolipram affinity are increased by phosphorylation of PDE4. It is, as I mentioned, a feedback mechanism via PKA, which increases enzymatic activity and the affinity of radioligand binding. As some evidence of this, we measured the density and affinity of PDE4 in rat brain. We did that before death using PET and a scatter plot, and then after death we did the binding studies. And what we found is that the affinity is decreased 5-fold after death, which is consistent with the known rapid dephosphorylation in presence of anoxia. So in the presence of anoxia, the enzyme becomes dephosphorylated, and many others too. So that would be consistent with it. We also gave local injections into the rat brain of drugs that either activate or inhibit PKA, and we got the expected effects on Rolipram binding. Taken together, this would mean that C11 Rolipram PET in humans provides a unique in vivo measure of PDE4 density and affinity, which is related to enzyme activity, that would not be possible in postmortem studies. So if you wanted to do these measurements postmortem, you couldn't because the brain is dead, the subject is dead, and the enzyme has been dephosphorylated. Okay. So this is the feedback mechanism specifically. Cyclic AMP acts at PKA. The A is for A in cyclic AMP, and which activates and phosphorylates PDE4. And what we can do is we can give drugs that will mimic cyclic AMP, like dibutyryl cyclic AMP, or we can give drugs that inhibit PKA, and we got the expected effects. So here's an example. This was giving an activator, a mimic of cyclic AMP, and you can see that we injected it on one side of the brain, and there was an increase compared to the contralateral side, as predicted. And someone might say, oh, well, that's just due to increased blood flow, because you activated the region and you just delivered more. Nope, because we also had the inactive enantiomer, the S Rolipram, and there was no such difference. This is a stereoselective effect of Rolipram. Stereoselective effect is on PDE4. And we can also do the other with an inhibitor. So this imaging evidence goes along with many other, with preclinical studies showing this inhibition. We were recently able to extend it to a rare human disorder called McCune-Albright syndrome. It's a rare mosaic genetic disorder, I think it's a single nucleotide polymorphism, of GS-alpha, so the G protein involved in the increase of production of cyclic AMP. It's a mosaic disorder. Some organs or some portions of organs are involved and not others. And we recently started this study, Laura D. Widener, who's in the audience, and we found the expected effects. So in these regions of the bone, which has fibrous dysplasia, there's a marked increase in the cyclic AMP production, and as expected, we see increased binding of Rolipram, consistent with the cyclic AMP stimulating PKA, increasing the phosphorylation. So we now have the first evidence in human subject that there's a similar sort of feedback mechanism in this rare genetic mosaic disorder. Okay, the sort of second portion of this is what is the effect of treatment. Chronic but not acute antidepressant treatments upregulate the cyclic AMP cascade. They upregulate all portions of the cyclic AMP cascade, including PDE4. And it's important to note that it takes weeks or months for this upregulation to occur. And that time course is very important because if you give an SSRI, the serotonin concentrations will increase within minutes, but you don't have an antidepressant effect until weeks or months. And so when preclinical animal researchers were doing studies, they were looking for biochemical changes that would mimic the time lag seen for the treatment of patients. And this was one of them. And so it led to the cyclic AMP theory as a common mechanism of antidepressant action was to upregulate the cyclic AMP cascade. And this chronic but not acute treatment has been shown in numerous papers by several investigators for all types of antidepressants, MAO inhibitors, SSRIs, etc. So chronic but not acute antidepressant treatments upregulate the cyclic AMP cascade, leading to the cyclic AMP theory of the mechanism of action of antidepressants. Mind you, prior to that, there's a lot of evidence suggesting the cyclic AMP theory of depression, that some types of depression are caused by low cyclic AMP signaling, and that this would reverse it and treat it. And we're going to be imaging the PDE4 with C11 labeled Rolipram. So the hypothesis going into this study is that unmedicated patients with major depressive disorder should have lower Rolipram binding than healthy subjects. The second is that the antidepressant treatment should increase the Rolipram binding in the patients. And the third would be the increase in the Rolipram binding should correlate with symptom improvement. For those who want to leave early, the answers are yes, yes, and no. Yes, we found that the unmedicated patients did have lower Rolipram binding, that antidepressant treatment for 2 months increased the binding, but there was no correlation between the increase in the binding and the improvement in symptoms. The subject group is shown here, and this is a good size. That's reliable, I would say. And these symptom rating scales here would be consistent, again, with, let's say, moderate levels of depression. And 23 of these patients had a follow-up scan. Okay, and the results were highly statistically significant, 0.01 level. Globally, there was in the major depressive episode, there was decreased Rolipram binding, consistent with the decreased cyclic AMP signaling and the cyclic AMP theory of depression. If you look at various regions here, it was true in all regions of the brain, including the cerebellum, 18% decrease at the 0.001 level. The global effect, I think, means that cyclic AMP may predispose, but certainly could not be the sole cause of depression. And this was global. We looked at it very carefully in different ways at the voxel and regional level. And I think what it means is you would have to have something like a two-hit model. A global decrease may predispose to depression, but must combine with many other parameters like decreased serotonin transmission to affect specific circuits and functions. I can say that a prior supervisor of mine would discount this and say that there's no utility to this if it doesn't involve specific circuits. And I would argue and say, no, that's just not true. Furthermore, it's not just a two-hit model. I've given this talk to people in psychiatric genetics, and there are tens or scores of genes that increase the predisposition to depression. Your type may be caused by 1, 2, and 7, and another one by 11, 18, and 19, etc. And some of those genes will have widespread effects, and some of them could be very specific and circuit-specific. But it doesn't mean that a widespread effect is not a predisposition to depression. Okay, so there was a widespread decrease in all regions of the brain of the Rolipram binding consistent with low cyclic AMP signaling. What happened after 2 months treatment with SSRI? So here's the treatment response. And this would be true for most treatment response in the United States. This is typical. 43% were considered responders, that is greater than 50% decline in the MADRS, and only 13% were considered remitters, decreasing the symptom scale of less than 10. It's these results that led me into research. This is horrible. I mean, we should be doing much, much better than that. And I don't feel like giving treatments that only have a 13% response rate. So that's why I went into research, because we need to have new and better treatments. Okay, so that's the symptoms. Now we look at the imaging. The imaging is shown here. The patients after 2 months treatment had an increase in their binding. We also imaged a group of healthy subjects 2 months apart. They weren't on any medications, and there was basically no change. So there's a chronic treatment causes an upregulation of the cyclic AMP system. But there was no correlation of increased binding with symptom improvement. And I'll come back to various explanations of that, potential explanations. Okay. In summary then, C11 roloprand binding in vivo reflects the activity of the cyclic AMP cascade in rats, and now I can say in a rare genetic human disorder. The roloprand binding was 18% lower in unmedicated patients compared to controls. The SSRI treatment increased the rolo binding in patients by 12%, exceeding the retest variability in healthy controls. However, the increased binding was not correlated with symptom improvement. The implications or impact of this study, one thing is that this study goes beyond measuring receptors at the cell membrane. It's one of the few that would look at a second messenger system. And it's the only one that I'm aware of showing that PET can be sensitive to the phosphorylation status of the enzyme. But as I mentioned, there was no correlation between symptom improvement and the increase in the roloprand binding. Why could that be? Well, one of the reasons could be variability. Variability in the disease and variability in the roloprand. So depression is clearly a heterogeneous disorder. Some caused by inflammation, some caused by cyclic AMP signaling, some caused by this, some caused by that. And with that great variability, in order to see an effect, you might have to have a much larger sample size in order to see it. Another problem is that roloprand is non-selective. It binds to all 4 subtypes of PDE4 in the brain. It's now known that there's PDA4A, 4B, 4C, and 4D. And the 2 most predominant ones in the brain are 4B and 4D. So maybe there's a selective effect on one of them. Bets are for the 4B. And it's being diluted. Okay. With regard to the impact, just going back to that, it is consistent with the cyclic AMP theory of depression and the theory of antidepressant action. So it also suggests that major depressive disorder could be treated with a PDE4 inhibitor. After all, if depression is caused by low cyclic AMP signaling, let's give a PDE4 inhibitor and it should treat the depression, right? Well, the cyclic AMP theory has been around for decades. And roloprand, that's why roloprand was developed in the late 80s and the early 90s by Pfizer as an antidepressant. And there were suggestive results that it was an antidepressant. It was stopped early after just a few days' treatment because of nausea and vomiting. I think it would be fair to summarize those studies as something like, well, despite the fact that I'm puking my guts out, I think my mood may be a little bit better. So they had to stop it. But I should say that that treatment response was within a few days. It was rapidly acting. Subsequently, it was discovered that there are these 4 subtypes, 4A, 4B, 4C, and 4D. The drug company said, oh, not a problem. We'll just make an inhibitor selective for one subtype, and if we have luck, it'll have efficacy without side effects. 20 years later, the first subtype selective inhibitor was reported by Mark Gurney, and that was for 4D in 2010, and for 4B by Gurney in 2014. So we've been collaborating with Mark Gurney to develop radioligands for them. We do have a good one now for PDE4D, as in David, and the protocol is under review and should be approved soon, and I hope that by October we'll be using this radioligand in human subjects to see whether it works. It'll also be associated with a occupancy study of a therapeutic candidate located at this site. So the idea of this global one of personalized medicine or precision medicine, whatever you want to talk about it, is that we can now use the PDE4 radioligand, go back to the depressed patients, is there a selective decrease or no decrease in 4D, and could you identify those that had the decrease and treat them with the 4D inhibitor? This is the image of the 4D radioligand in monkeys, and we hope to start in human subjects in the fall. So towards my overall goal of trying to link PET to treatment trials, I think there are two basic ways in which it can be helpful. One is patient stratification, and the other is measuring drug delivery to the brain. When I was in medical school, it was just called patient stratification, and that is you want to try to find that subgroup that's going to respond to your treatment. It later was termed personalized medicine, and I hated the jargon. But after about a decade, I just started to use it, and then it's been changed. It's now precision medicine. And even President Barack Obama uses the term when he's come to NIH for a backdrop to give a press release. So I guess we're going to have to call it precision medicine whether we want to or not. But the basic idea is to stratify the patients into those that we think should respond and those shouldn't respond. So for the inflammation, find those with high inflammation and low inflammation, treat with an anti-inflammatory, and one would predict a greater response in the high group than in the low group. And if so, then we really do have a biomarker that has utility because it's guiding treatment. In a similar sort of way, we can use roluprem or some C11 PDE4 radioligand to identify those with high and low cyclic AMP, and then treat with such an inhibitor. And if this radioligand for 4D works in humans and it can be occupied with reasonable doses of the therapeutic candidate, we actually plan to do such a target treatment. With regard to drug delivery to the brain, this is a very important issue for pharma. How do you know that your drug actually got to the target, engaged the target? Because if you end up with a study, a $200 million study, and you find there was no effect, is that because maybe the drug just didn't get to the brain? So in years past, when Big Pharma was involved in psychiatric research and development, by the way, almost all Big Pharma has given up on psychiatric research and development. They would require that when you start a new target, you have a radioligand for it. And this is an example of a target that, this is the latest radioligand that we've developed. It's in collaboration with Lilly for a compound called OGA, oglycanase more formally. In any case, this is an enzyme that if you inhibit this enzyme in rats or mice, you increase the clearance of tau protein from the brain, and the mice improve their cognitive performance. So in anticipation of future treatment trials with this drug that would remove amyloid, remove tau from the brain, we've done studies like this, and you can determine, with whether there's partial or complete blockade for various doses. We have actually are almost finished now with the human studies, and this radioligand looks very good, and it will be helped to guide the doses in any future treatment trial that Lilly takes on. Okay, so that's the idea, using the PET imaging to measure target engagement and also for patient stratification, those with high or low inflammation, high or low cyclic AMP signaling, and then treat with the cognate thing that's really directly related to it. I want to thank several people, but particularly Victor Pike, the director of radiochemistry, and I'm happy to take questions as time permits. If you could, I was asked to try to use the microphone because these are being recorded and stored, but I can also repeat the question. Yes. Oh, no. Nora. Okay, one of you. Who's going to ask the easier question? Okay, good, I'm glad. Nora always has the tough ones.
I was just curious, thanks a lot for the talk. With the
Could you speak up?
Louder? Or?
Maybe turn it on.
Okay, I think it's
Whatever.
on. All right. So the question was there any correlation between TSPO binding and levels of beta amyloid in the study that you looked at?
I didn't hear you the second half.
Was there any correlation between TSPO binding and beta amyloid levels in the study that you reported for the Alzheimer's?
No, there's not a correlation with that. And in fact, one would not expect it because amyloid reaches a peak at MCI and stays the same. The TSPO is increasing. The correlation was with clinical impairment and also the colocalization with tau. So tau protein and this inflammation marker correlate with disease severity and are co-localized together. You don't know which one causes which. I mean, it could be the deposition of tau induces an inflammatory response, or vice versa, or it could be a combination of the two. But it's tau and inflammation, not amyloid and inflammation.
Okay, thank you.
Oh, thanks. Yeah, no, but first question, and you say since you are expecting very difficult, will you give, I mean, fantastic presentation, so will you give access to the PDF files of your presentation?
Oh, no. Of course. I have a better one because there were some mistakes I noticed in here, but I'll send it to you.
Yeah, no, it was wonderful. And my question is, I mean, certainly, I mean, one would want to see a one-to-one pattern, but you are basically making your measurements at a very precise point in time. So I was wondering to what extent, I mean, you could, by looking at trajectory of depression, for example, start to distinguish what that significance may be about why is it that you are observing an upregulation in all of them and yet not associated per se with the improvement. Does it have any, on the other hand, for example, in other words, correlation with their prior history of responsiveness to treatment? Is it in any way related beyond that moment where you are doing the clinical evaluation?
We didn't have enough sample to do that, but Jeff Meyer, who did the first such study from University of Toronto, has published a much larger one now. And the correlation he finds is that the increased TSPO binding was with the duration and severity of depression, or lifetime. So it's really consistent with the idea that this TSPO is very slowly responsive, sort of like the hemoglobin A1C, but maybe even longer. So it may be a better marker for those of the chronic inflammation existing in the weeks or months previously. So the study that I would like to do, and Jeff might collaborate with me on it, is let's use this TSPO, this hemoglobin A1C, to identify those individuals with high and low, chronic or not so chronic inflammation. And then the treatment that I want to give is actually with a COX-1 inhibitor. There's increasing evidence that, that in the brain, the bad player is COX-1, not COX-2. And that would be suggested by the cellular localization where COX-1 is almost exclusively in the microglia. And a good example of that would be studies, and the utility of PET was that for Alzheimer's disease, there's been a long interest in anti-inflammatory treatments. It goes back to the 1990s, when there were very large epidemiological studies, particularly from the Netherlands, but others, showing that decades-long treatment with NSAIDs significantly reduced Alzheimer's disease, significantly meaning by about 50%. So given the fact that there's this post hoc epidemiological data that it can be protective or preventive, NIH funded a very large study in something like 1,700 patients, 1,700 controls, and they gave a COX-2 inhibitor, celecoxib, and it had no effect whatsoever. Then subsequently, someone measured the CSF concentrations of the celecoxib, and it was very, very low, probably too low to be therapeutic. So one of the utilities of these COX-1 and COX-2 radioligands, it will be able to determine what dose do you really need, does it get into the brain, and how much. I can say that there was another study done that had to be stopped early, and that was with Vioxx. So Merck also did a study with Vioxx. Vioxx, in contrast to celecoxib, has active entry into the brain, but they had to stop that study early. There were only 1,000 patients and 1,000 controls, and they found it had a statistically significant negative effect. So the COX-2 inhibition was negative in the brain. The study had to be stopped early because Vioxx withheld information about the cardiac cytotoxicity, estimated to lead to tens of thousands of excess cardiac deaths. In the class action lawsuit, Merck had to pay $3 billion, but you know, the sales of Vioxx was $3 billion a year. It's just the cost of doing business. But I would go for COX-1 inhibition in the brain. So what I'd like to do is do the TSPO high and low, using our we have a new TSPO radioligand. Treat with a COX-1 inhibitor and see if there's any response and a differential response. Because then you'd really know whether this TSPO is a useful marker. I should mention that there's a lot of controversy about how useful TSPO is in various disorders. And it has to be proven, I would say, in any particular disorder. But in the end, the real proof is going to be, in my mind, if it can be linked to therapies. Adam.
Really exciting talk, Bob. I enjoyed that a lot. So the PDE4B and the PDE4D, was it?
B and D, Bob and David.
Bob and David, developed in 2010 and 2014? Seems like a long time. Do we have any indication as to whether they're going to be therapeutic or not?
The animal studies, for what they're worth, suggest that inhibiting 4B will be improve cognition, and inhibiting 4D will improve, no, 4B will improve depression, 4D will improve cognition.
And the nausea?
It's unclear, but probably 4D is the one that's causing nausea, but not clear. So we're trying the 4D that is thought to improve cognition, and we're going to be trying it in depression. Well, there's also a cognitive impairment in depression, and that would be worthwhile. And also because the animal studies, you can't trust them anyway. You know, you can't be sure an animal really has depression. And I can say that that has been the 4D that we'll be using here for blocking the radioligand has been tried in healthy subjects, and believe it or not, it had a statistically significant effect in healthy subjects of improving cognition. But my hope for depression would be for B, and, but that, to my knowledge, has not yet been administered to human subjects. One last question. Jean-Jacques Wang.
Hi. It's very interesting. In the beginning, yeah, you talk about is that a lot of tau accumulation in posterior cortex.
In posterior cortical atrophy, in a
Posterior
rare variant of
cortical, yeah.
Alzheimer's
Okay.
disease.
So do you have any follow-up about that kind of study that is that this location will gradually merge into other location of the brain, such in the frontal cortex? It's a different kind of disorder, right?
It's considered a variant of Alzheimer's disease in which the primary pathology is in the occipital cortex with a deposition of tau there, and with our studies, also this marker of inflammation. It does progress, but I don't know the full extent of how far it goes into the frontal cortex. There's another disorder, dementing disorder, where it begins in the frontal cortex, frontotemporal dementia, FTD, and a paper that we have in preparation now. As expected, for the frontotemporal dementia, we see the inflammation in the front. So we have two sort of rare disorders where you see it in the appropriate region, and then Alzheimer's disease where you see it everywhere, basically, except the cerebellum. And we're going to be studying more of these rare patients, particularly frontotemporal dementia, largely coming from UCSF. They have the largest behavioral neurology clinic and study of these the frontotemporal dementia genetically caused. So we can image the gene carriers, some of whom are affected, some of whom are not yet affected, and siblings and stuff. So it's it's a horrible disorder, but it's very useful for these sort of studies we're doing to see the utility of PET inflammation in this disorder. Thank you very much.